today we will be going over amino acids, and I believe that amino acids are one of the most important subjects in biochemistry. So please pay attention, take notes, because you will use amino acids a lot in biochemistry. It's one of the foundations of every process that you'll see from now on. Okay, so we'll begin with describing an amino acid. Okay, so what is an amino acid? Well, first of all, there's about 20 naturally occurring and amino acids from now on will be referred to as AA, okay? So naturally occurring amino acids, and well, in their natural form, so if you were to purify it, they would actually be these white powdery solids, okay? So imagine like powder, like flour, okay? When you bake with flour, it's the same consistency of a flour, okay? So it's a white flour-like substance, okay? And it's not going to melt at room temperature. You have to have like a very high temperature to melt it. So what do amino acids do? Well, amino acids can act as enzymes, meaning they could speed up reactions, aka they're catalysts, and they can manage waste. They can actually create energy as well. So we can put carries. We can put carries energy, and also carries waste. Okay, they make up hormones, or they can actually carry hormones. Sorry. Ugh. And sometimes they can act as intermediates in a reaction. We'll just put RXN for reaction. Now, proteins actually make up, so amino acids actually make up proteins. So amino acids make up proteins. And the way this happens is that you get an amino acid and you're going to link it with another amino acid. So individually, if you just have one amino acid, that's a monomer. But when you connect it to another amino acid, you get a polymer. In this case, the polymer would be called a protein. Okay? So what is this? This right here, this bond that links them, is a what? It's a peptide bond. So you're probably wondering, okay, well, that's nice and all, but if there's 20 amino acids, how do I, tell which one is which? Well, amino acids have different structures, and those structures, well, they designate the function, how it reacts with certain compounds, the name, or just kind of like the identification of the amino acid, okay? So in this case, you can say that, because naturally amino acids, they typically have something like this, where we have H2N attached to a carbon, attached to COOH, attached to hydrogen. And then this is where it gets a little weird. Okay, this is a little weird. You have an R group. Now remember in organic chemistry where the R group could be like a series of carbons or whichever, like R's can be almost anything. Well, the green part is always going to show up in amino acids. That's what it makes an amino acid, right? The only thing that changes is the R group. Okay, you might have a carbon there, you might have an oxygen there, or an alcohol, and that is going to change the whole structure. It's going to be a different amino acid. And so you can technically say, well, you can say that amino acids always have an amine, which is this part right here. That's an amine or amino, and it's always going to have a carboxyl group. So that could change. This is a carboxyl group. The only thing that really changes is the R group, okay? Alright, so now what do we do? Well, what happens if the amino acid undergoes a reaction? Meaning, like an acid-base reaction. So if that happens, this hydrogen can actually leave. So let's draw a bond with the hydrogen. It can break out, and this nitrogen, which has that right there, can actually pick up the hydrogen, okay? So let's see, 2, 4, 6, 8, okay, yeah. And when that happens, you actually get a really cool product. And let's just kind of make a red R, okay? So when that happens, you actually have what? You have an acid and a base on the same compound. That's really weird. So in some textbooks, you might see the amino acids written out as COO and H3N. Sometimes you might see H2N or COOH. Well, that's just how they prefer it. So whichever they prefer, you might see that. But on amino acids, they actually have a base and an acid on the same compound. However, this actually occurs only at the pH of 7.4, which is the pH of blood. So occurs at a pH of roughly 7.4. Now, this whole compound, in general, whenever you have a structure that has a base and an acid at the same time, that is called a Zwitter ion, okay? So a Zwitter ion is a structure that has both a base and an acid at the same time. Now that only happens to amino acids at the pH of 7.4, which is the pH of blood. Of course, as in life, there's always going to be an exception. So what is the exception? So the exception is with proline, which is an amino acid, and we're going to talk about proline later. But all you need to know is that I'll just write it out. So here's proline, okay? Here's what happens with proline. Okay. So that's proline, okay? Now notice that nitrogen doesn't have any has any lone pairs to give off, okay? So all its lone pairs are being used to form bonds. So it's not going to be able to take away this hydrogen on the carboxyl group, okay? So that's proline, and that's going to be literally the only exception to this rule. So it's not going to form a Zwitter ion. It doesn't matter if the pH is 7.4. It's still going to maintain its rigid shape. Okay, and proline is going to make another showcase whenever we're talking about alpha helixes. Okay, so once again, proline, because of its structure, does not allow the nitrogen to grab the hydrogen from the carboxyl group. And just like a quick just like a quick mention, sometimes textbooks are actually going to omit the hydrogen, okay? And you might see it like this, which is fine. Just make sure that the hydrogen that left does not, and I repeat, does not go to the nitrogen, okay? It might go to some other compound or whatever, but depending on how you write it, make sure that you do not have H3N for proline. You have H2N for proline, okay? Now, sometimes, well, actually all the time, amino acids exist within the stereochemistry universe, okay? So what I'm trying to say is amino acids actually have stereochemistry. So amino acids have stereochemistry. If you haven't taken organic chemistry in like a couple of months, or you are taking it at the same time, stereochemistry just means how the structure looks like in 3D space, okay? So sometimes, you know, the hydrogen might be going towards the back and not the front. Well, if it's going towards the back, it's going to be a different compound. If it goes towards the front, it's going to be a different compound, okay? So amino acids have stereochemistry, all right? And it's pretty obvious of how you name it or how you look at it, okay? So first of all, we go by weight, so molecular weight, right? So we start at this alpha carbon. Now the reason why this carbon right here is the alpha carbon is because it is next to the most electronegative atom. So here's the oxygen, which is the most electronegative. So here's the parent carbon, and the carbon that is next to the parent carbon is the alpha carbon. So the alpha carbon comes after the parent carbon, and the parent carbon is right next to the most electronegative element or atom, okay? So from the alpha carbon, we start looking at what weighs the most, which has the most weight, okay? So we're looking at nitrogen, hydrogen, carbon, or carbon, okay? So you're probably saying, okay, well, you know, this is number 1, and that's number 2, and this is going to be number 3. That's wrong. It's wrong because you said, okay, well, this carbon has 2 oxygen. That's really heavy. That's number 1. No. No. You look at the individual atoms first, okay? So really it's between nitrogen, carbon, hydrogen, and carbon, okay? So nitrogen is going to have more weight than carbon right here, okay? It's going to have more weight than this carbon, and for sure it's going to have more weight than this hydrogen. So we're going to call this number 1, okay? So now I go down the line. Now it's either between this carbon, this hydrogen, or this carbon. Well, this carbon doesn't have any hydrogens, okay? So that looks pretty good. And, well, since it doesn't have any hydrogens, it's automatically the best one, right? So this is number 2, right? Well, okay, there's a trend, okay? So if you have CH3, well, that is not as good as H2C, and that's not as good as HC, and that is not as good as carbon, okay? So this is preferred. Ultimately, whenever you're doing the numbering, okay, for stereochemistry, you want to have carbon by itself, okay? So there you go. So here's an example. You have a child, and you don't want that child to eat 3 hamburgers, right? You don't want that. You don't want the child to eat 2 hamburgers. You don't want the child to eat 1 hamburger because it's past their bedtime. So you just want the child by itself to go to sleep, okay? Babysitting is a hard job, I get it. So that's one of the examples right there. This child doesn't have any hamburgers, it just has oxygens, so that's going to be the best one. So the rest, obviously, is going to be number 3, because carbon weighs more than hydrogen. This is going to be number 4. So we go from 1, 2, 3, and 4. That's really like this, okay? But notice that the Number 4 is going out towards the front. So whenever you have that, you would have to reverse what you did. Okay, so you go this way. And whenever you go that way, okay, that is an S configuration. So that is a silly way a clock moves. So silly way a clock moves. And S just means that it goes counterclockwise, okay? So counterclockwise. That's an S configuration. And we will talk about more about S and R configurations in a couple of minutes, because in biochemistry there is a special denotation. So this is actually S configuration, okay? So to recap, amino acids have stereochemistry. In this particular example, this was an S configuration. Also, if you're curious, this is actually alanine. Okay? And if you want to be more specific, this is L-alanine. Now, here's a fun fact, and it might be on the exam. It usually is on exams. Well, you know, you only use L amino acids to build proteins. Okay? And we'll talk about that later on in this video. So now we will talk about the denotations within amino acids. Okay, so you remember how I just talked about S configurations and R configurations? Well, in biochemistry, they go a step further. And really, it's the same thing, but with different letters, okay? So let's begin by trying to figure out the direction of which this structure bends light, okay? So that's what you're doing, okay? So let's start by the alpha carbon. Okay, let's start by the alpha carbon, and of course, they prefer to use the acid and bases connotation. Okay, so you don't have a hydrogen right there. That's perfectly fine. Okay, so we'll start. So nitrogen is going to have a higher weight than this carbon. Okay. Now this carbon has less hydrogens than this carbon, so it's going to be number 2. And this is going to be number 3 because it's better than hydrogen. And that's going to be number 4. So we go 1, 2, 3, and 4. We go this way. But notice that my lowest group, sometimes it's not even hydrogen, it just has to be the lowest weight, okay? So the atom with the lowest weight is on a wedge. Therefore, I have to reverse what I did and go this way. So that is counterclockwise, which means that it is S, okay? So now you know that S is equal to what? L. Okay, so in biochemistry, you don't say S-phenylalanine, you say L-phenylalanine. And you also know that whenever it's L, it's going to be the preferred amino acid to build proteins. Another way you can think about it is that if it goes towards the left, if it goes towards the left, then it's going to be negative, okay, because it's counterclockwise. That's not the good way a clock works. If it goes towards the right, we'll call that D, is going to be positive, okay? And of course D, or let's say R, is equal to D, right? Now the reason why we have S is equal to L and R is equal to D is because S is it's actually left-handed, and L means lexo, okay? So lexo in Latin means left-handed, and R, which is right-handed, means what? Means what? Dexo. Or rather, dex, okay? So dexterity is right-handed, and also this is actually levo, not lexo. As levo. So levo is Latin for left-handed, meaning that it goes counterclockwise, and R, which is right-handed, means dex, which is what? Right-handed, where it goes towards the right. Okay? So there you go. So you can think of it as S-phenylalanine and R-phenylalanine. Of course, in biochemistry, you use L and D, right? Okay. So if you can't remember, just make an L with your left hand and you say, hey, that means that it goes towards the left. It's negative because if a clock moves towards the left, that's a bad thing, okay? So there you go. Let's just kind of do this one right here. So that's going to be 1, 2, 3, and 4. That goes 1, 2, 3, and 4. That goes like that. Well, the hydrogen is in the front, so we reverse it. It goes towards the right, and when that right is equal to D, which is dex. So that's right-handed, okay? And you only use the L's to build proteins, okay? So you can actually buy L-phenylalanine at the pharmacy and use that to build proteins in your bodies. Okay, so what? Why is direction so important in biochemistry? Well, direction is really important because depending on what direction the substance is going, there will be different effects. So for instance, this is actually carvone. So this is carvone. Okay, and this is also carvone. So let's figure out what direction it's going in. So we start with the alpha carbon. So this is going to be number 1 since it doesn't really have that many hydrogens. Okay, it only has like 1 hydrogen. So that's pretty good. Now this, whenever we're counting, we go down the line. So now we're focusing on these two and this guy. Okay, so these have 2 hydrogens and this doesn't have any hydrogens. So that's going to be number 2. And now we're focusing on him, this guy, and this guy. Alright, well that one only has 1 hydrogen, this one has 0 hydrogens, that has 3, that has 2, okay? So obviously this guy is going to win, right? And then we go back to whatever, so it's going to be like 1, 2, and 3, so it doesn't matter. We go 1, 2, 3, okay? Well that's going to go in the opposite direction, so that's going to be L, okay? So that is S. So we call that L, so that's L carvone. We can also call that negative. All right, let's do this. So that's negative carvone. And if you were to do the same thing, we say 1, 2, 3. We go 1, 2, and 3 that way. Well, that's the correct way a clock works. So it goes to the right hand, and so that's D. So this is D carvone, and you can also call that a positive because that's the correct way a clock moves, okay? So who cares? Why does it matter? Well, carvone is actually a substance that has flavors, okay? So the negative, so the L carvone actually has a minty flavor, so that's actually spearmint, spearmint gum. Okay, so whenever you bite into gum, you're most likely tasting negative carvone or L carvone. Okay, so what is the other one? The other one will actually have to be caraway. Okay, so that's caraway flavor. You're probably wondering, what is caraway? It sounds disgusting. Well, actually, it's not that bad. If you ever had pizza, and I know I have, yeah, you often see these little herbs on the crust, and whenever you bite into it, it's kind of like an oregano flavor. Okay, it's kind of it lingers on your tongue and is very herby. Okay, so that's kind of like pizza flavor. So it's like pizza flavor. Okay, so you can see that if they really wanted to make pizza flavored gum, all they have to do is just reverse the direction of the substance, okay? But I don't think that's going to sell well, so they stick with spearmint, okay? So what happens if, these substances aren't as harmless as flavors? So sometimes whenever a pharmacist is making a drug, let's say like a morning sickness pill, they typically want to have the direction that doesn't kill you or provide any side effects, okay? So for instance, there was a drug aimed towards pregnant women, and it was called thalidomide, okay? And what happens was, this was like a morning sickness medicine, and the R, it was good. R was good. Okay, so the D was the preferred direction, but the L actually caused birth defects, okay? So you would have babies having their foot split in half, and they would have kind of like these weird appendages, and it was not good. That was during the 50s and 60s, and they said, hey, we have to limit how much L is in this medicine. So they got it down to like a very, very small amount, okay? So to this day, whenever people are making pharmaceutical drugs, they try to purify the compound so that it gives them the correct direction, okay? However, if the opposite direction is harmless, then they allow a 50-50 mixture to go into the drug. It's not going to kill you, it's not going to do any side effects, just keep it in. It's more efficient to just keep it in. However, if it could kill you, there's going to be like a 99.9% chance that it's only going to be the good stuff and not the bad stuff, okay? So whenever you're taking L-arginine, if you're building muscle, you can expect about 99% of that powder to be the L direction and not the D direction, okay? So stereochemistry is very, very crucial and vital in biochemistry, okay? And finally, sometimes the amino acid in your body, as you age, will actually convert from L to D or D to L. So amino acids over time can convert to the opposite direction. And so you have all these Maybelline companies that try to reverse the aging process. So they're trying to find a substance that will try to reconvert the L from the D, right? So they don't want this substance to convert direction. They're trying to keep the direction as normal for as long as they can. So that's called the anti-aging cream. All right, sometimes it works, but it's not going to be like a miracle cream. All right, we're not there yet. So main takeaway is that amino acids over time in the body can change directions and when they age, okay? Now we will be starting our discussion on amino acids in very minute details, okay? So this is when we actually start drawing out each individual amino acid and assigning its one-letter abbreviation, some of which don't make sense, and the three-letter abbreviation. So three-letter abbreviation is very important whenever you're reading a textbook or an exam and they say, describe ILE, and you're like, okay, well that's isoleucine, right? And the one-letter abbreviation is just I, right? So I recommend that you just pause the video and copy this down for reference, and then we're gonna go into the three main groups of amino acids, okay? So the first group that we're going to be talking about are the non-polar groups. Non-polar amino acids, aka hydrophobic amino acids. So these are the guys that only have carbons or something that does not react readily with water or other polar compounds, okay? So these are going to be pretty basic molecules. And by basic, I mean simple. We will now begin drawing them out. Okay, so these are, it's going to take a long time. So it's H3N. Here's a carbon, and now here's a COO negative. So whenever I'm drawing these out, I'm going to have the acid and the base within the substance, okay? So here's the H right there. Now, this is where it gets a bit different, okay? This is where it gets a bit different. Over here, we have, instead of the R group is a CH3, Okay, so notice if you look back on your notes, I said, hey, there's an R group, and this R group can change, and depending on what it changes to is going to be a different amino acid. In this case, the R group is converted to a what? A methyl, right? Or methyl. So that is just alanine. So alanine, right here, we're gonna, I guess, put over here. So that's alanine. It's pretty simple. You could put al, right, and also a. So that is alanine and a, right? Okay. So what happens if we make its brother? So whenever I'm drawing these out, I'm trying to make connections, okay? I'm trying to make the simplest connections that I can do, okay? Now, here was alanine, right? Here was alanine. But what happens if I add something else? What happens if I did this? Right? Well, congratulations, you actually made valine. Okay, so valine is the brother to alanine. So val, ene, which also goes as val and v. Okay, well, what if I make the brother of valine? Okay, so what if I did this? What if I did it over here, actually? H3N plus, then there's a carbon, there's a COO negative, here's a hydrogen. Okay. Now, what if I did this? Okay. That was a bad H. And we go CH3, CH3. Well, right there you make leucine. Make leucine, which is LEU, and also L. So you can see kind of like the difference of the compound. So if you just start from alanine, okay, well, we just add a methyl right there. Okay, well, what happens if I added 2 more methyls to that methyl? Well, you're going to get a valine, right? All right, but what happens if I add a methyl to this guy, and then I just move the methyls to that added carbon? Well, if I do that, I'm going to get leucine. Okay, I'm going to get leucine. Right? So that's kind of like the thought process you want to have. Hey, what is the pattern that I'm doing? How can I make this simplified? How can I make it more simple? Okay. Well, what would happen, for giggles, for jokes, what happens if I move this guy over here? Huh, interesting. Let's figure it out. What happens if I did that? Well, if I did that, I would have H3N plus, then the carbon, then the COO negative, then a hydrogen. Okay, and let's add this right here. So we have CH, okay, because we're adding something to that carbon. We're gonna have CH3 right here, and then we have our carbon with what? The CH3. Whoa. Okay, so the CH3 right there. So 1, 2, 3. Right? So that's going to be CH2 right here. Okay, so 1, 2, 3, 4. So that carbon has 4 bonds. Alright, so let's recap for the first 4 amino acids, okay? I know it seems excessive, but really get it in your head. So we start with the basic amino acid, and it's the most simplest. You just add a methyl, that's alanine. Okay, well, what happens if you add 2 methyls to it? Well, you're going to get valine. And what happens if you add another methyl and then shift the 2 methyls from the first carbon onto the added carbon? Well, you get a leucine. Okay, cool. But what happens if you shift 1 methyl to the previous carbon? Well, if you do that, you're going to get what? You're going to get isoleucine. Ooh, isoleucine. Okay, also known as ILE. I don't like writing the L in lowercase because it looks like another I. I don't like that. So there you go. And the single letter for that would be I, okay? So these are all just derivatives, and derivatives just mean that they're kind of like the next step in evolution. So they're all siblings, okay? You got al, you got val, you got lu, and you got isolu, alright? So isolu is kind of like the little brother for lu, and they're all just one big weird family. Now there's also some other amino acids that are still hydrophobic, and we're going to talk about that right now. Again, the most simplest way to remember this next one is to go back to your simplest compound, alanine. All right, what happens if I write it again? Okay. Well, I have a carbon. So that's pretty simple, but what if, just for giggles, I do this? Right? And then I do something like this. Look at that. So this would be actually a CH2, right? And, well, what is this compound right here? Well, it's directly attached to a carbon, so it's a phenyl. Okay. And if we ignore everything to the right and we just focus on the left, that kind of looks like alanine. So common sense says, hey, you know, maybe this is called phenyl alanine. What? Yeah. Everything kind of builds up on each other, and it's not that difficult for at least like 5 of them. Okay? This is pretty simple. That's phenyl alanine. And the 3 letters would have to be P, H, E, and fifth, like the PHE sounds like F, right? So that sounds like F. So it goes F right there. So phenylalanine, PHE, F, okay? So again, phenylalanine, you're just adding a phenyl to the alanine, right? And just make sure that your carbon always has its 4 bonds occupied, okay? Or I guess always has 4 bonds. Now, the next couple of ones were pretty difficult to remember, but there's only like 3 left, so here we go. We have our basic structure right here. And then we go to the hydrogen. Don't like that. Ugh. So, of course, we're going to add the thingy, C, right? H2, because we're going to be linking something else. And then we're going to say, okay, well, what if we had another CH2? So notice that I started with the alanine. Well, what if I added another carbon? Okay. But now this is where it gets a little weird. Okay. You're going to add an S. You're going to add an S, which is sulfur. And then you're going to top it off with CH3. So the main thing that you should remember for this compound is that there is an S. So that's a sulfur. Okay, so this is called methene-O-nine. So that is methionine, okay? Methionine. Why is it called methionine? Well, look, there's a methyl group right there. So that's a methyl group. Okay. And then there's a thio. So thio is kind of like shorthand for sulfur. Okay, so that's a thio. So it's pretty cool, but rather there is there's a methyl at the end. There's a sulfur, and the sulfur is about I guess you could say it's like the second to last atom in the structure. Okay, so that's what it's called. It's not that intuitive, I would have to say. Methionine. However, the name is pretty intuitive. So MET right there, and the first letter, M. As you progress, those names are not going to become intuitive. They're going to be weird, okay? So I'm going to clear the board because everything is going to go topsy-turvy for the last 2 nonpolar amino acids. So the next one is like literally the hardest thing to draw for the hydrophobic amino acids. Okay, so we first start with CH2. So you're like, okay, well, it looks like alanine, but then it kind of trips you up. Okay, because then it takes a literal nosedive right here, and we have NH, H right there. We go over here, that way, and then we're gonna draw a hexagon. Okay, so we're gonna do this. So that's 1, 2, 3, 4, and 5. That goes here. Boom, boom, and boom. And of course, this is called tryptophan because it tripped you up on how to draw it. And that's called TRP, which is trip. And it's a W, a major W, big dub, if you actually draw it correctly the first time. So hopefully that actually helps you memorize the name. So it's kind of weird how it looks like, all right? So from what I gathered, is that you want to start with the basic, simplest amino acid structure, and then you're going to draw CH2, almost like an alanine, but then you're going to draw a pentagram or a pentagon, except this point is not a carbon. That point is actually a nitrogen with hydrogen, okay? Now, towards the 90-degree angle or whatever, you draw a double bond, okay? Then you go to this carbon, you draw a double bond right there to make it resonance, okay? To make it aromatic. So that's essentially what you want to have. You want to have an aromatic compound, okay? So that is tryptophan, TRP, and that is W, okay? And the other one is not too bad. The other one is like this. You already know what it is. Proline. Actually, if it's proline, here it is. You're going to have carbon, COO8. Yeah. Yeah, you could do it. You can have that. So this is what happens right here. You want to have this guy and this guy combine. Okay. So let me see if I could draw that better. And there is an H, okay? So I probably should have done that in a different color. So if you'll excuse me, I'll do it in a different color. So first of all, since this is proline, you do not want to have H3N. You actually want to have H2N, okay? So we're going to eliminate this. We're going to have H2N in a different color. So that's H2N. And then we're going to have a bond right here. And there you go. So not too bad, actually. So because of nitrogen's bond, it doesn't have any electrons to grab the hydrogen from the carboxyl group, and therefore the hydrogen is going to go somewhere else, right? So this is proline. This is proline. And remember that you have to be a pro to remember not to have H3, but rather H2, and that is a very simple P. So it is pro and P. Now we will be talking about polar amino acids. It's not too bad. About 4 of them are very similar to the hydrophobic amino acids, okay? So first of all, we will always begin with our simple structure for amino acids. Okay. Now, instead, so remember alanine, right? Remember alanine. Remember how we used to put CH2, right? If we're gonna, base something off of alanine. Normally it's H3. But if we do that, and then we add something a bit special, if we add an alcohol, you actually get a polar substance, okay? So that polar substance is called serene, because it's very calm. It's taking a deep breath of relaxation. Oh, I'm calm. Okay, so that's serene, serenity, serene, and that's called SER. And S for the single letter term, okay? Likewise, let's try to make another kind of like example out of this one. So let's try to make a derivative, or the brother of serine. So that's H3N plus, we have a carbon, and COO negative with the hydrogen. Then we're going to have our CH2. But instead of adding an alcohol, instead you're going to be adding what? Sulfur and what else? A hydrogen. So we have SH. And this is called CYS-tene. So that is cysteine. The three-letter abbreviation is CYS, and the single letter is C. So remember, so look at it. Instead of having the alcohol, you actually have the SH group. Okay, so that is called cysteine, CYS-C. And likewise, let's try to recall phenylalanine. Okay, let's recall that. So let's draw the basic structure. Okay, we have something like this, CH2, but instead of just having the phenyl, we're going to have this. So let's draw the hexagon. Okay, and we do this, boom, boom, and what else are we going to do? We are going to add an alcohol directly opposite to the CH2, okay? So that right there is called tyrosine, okay? And the three-letter abbreviation is TYR, and that is just a Y, okay? Because the Y comes out whenever you say tyrosine, okay? So just remember that it is tyrosine, this TYR, and the Y is a single letter abbreviation. It is based off of phenylalanine. Instead of having just a phenyl, you're going to add an alcohol or hydroxyl group to the opposite side of the carbon. Now let's remember valine, okay? So valine was like this. CH, and then we're going to have a carbon right here. Okay, but what is the difference? Well, the difference is that instead of having another methyl group, you're actually going to have an alcohol group, okay? And that is called threonine. And threonine, abbreviated, is THR or T. Okay, pretty simple. So you just remember valine, you take out one of the methyls and you add a hydroxyl, and that is going to be threonine. Now we're going to do some weird polar substances, okay? So of course we're going to start with CH2. Then we're going to do another carbon, but this carbon is a bit different. This carbon's a bit weird. It is actually doubly bonded to an oxygen, and afterwards it has NH2. So this one is called what? Asparagine, okay? It's always quite hard to spell, but it's asparagine. And of course I misspell it because, yeah, sorry, so asparagine or something. Okay, so that is actually ASN, also known as N. So how do you remember this? Well, you remember it via the ASN. Well, the single letter is just going to be the N again. Okay, so the N again is the letter for asparagine. So it rhymes, I guess. So just remember that you're going to start with your basic structure for an amino acid. You add your CH2, you add a carbon, but then you're going to add an amino group. So that's what the INE is for, amino, right? So right here you have the carboxyl, or I guess you just have a double bonded oxygen, and then you're going to be bonded to an NH2, right? So that's going to be called asparagine, ASN or N. Now let's do the brother to asparagine. It's going to be this. And let's do it again. So it looks pretty familiar, right? But what if I did this? And that is called glutamine. So that looks like an A. Glutamine, also known as GLU, sorry. So let's rewrite that. Okay. And now here's the kicker. The letter actually is Q. Yeah, I know. So glutamine apparently has a Q in it. Don't know why. And it's GLN. So glutamine. It's either glutamine or glutamine, whatever you like to call it. Okay? So those are going to be brothers, they're siblings. And in the next section, you're going to see how they evolve directly. Okay? But there's only two more polar amino acids left. Okay, so you start with your basic structure for an amino acid right here. And now we start doing some weird stuff. We're actually going to make a pentagon, but you'll see how it's weird. Okay, so we're going to do this. That's 1, 2, 3. So instead of a carbon, this is actually a nitrogen. Okay, then we go downwards. This is going to be another nitrogen. And then we just bond it like that. And right here is going to be a double bond here too. So let's do that better. Okay. And there is a hydrogen. Okay. So let's see. First you start with your simple structure for an amino acid. You add your CH2, and then you're going to make a pentagon. But instead, across from each other, there's going to be a nitrogen. Right? Nitrogen by itself and nitrogen with a hydrogen. The nitrogen by itself is going to have a double bond going downwards, and then you're going to skip a bond upwards and add a double bond right there. Okay? And this is called histidine. Okay? And the 3-letter abbreviation is his, and the single letter is H. Finally, we get to the last one, the last polar amino acid, and it's very, very simple. Okay? So here it goes. You ready? It's another H. Okay, there you go. So it looks kind of weird, but you're probably wondering, how's that polar? Well, that H can actually deprotonate and become a polar substance, okay? So that is called glycine. And you spell it like this, glycine, and also G-L-Y and G right there, okay? So that's glycine, G-L-Y, and G. It's the most simplest polar amino acid. Actually, it seems I made a mistake. I don't know why I thought this, but actually this is a non-polar compound. I guess I forgot to do it in like the first section, so I apologize for that, but this is a non-polar substance. Okay? So it's a non-polar substance right there. So glycine. Now we will be talking about the charged amino acids. So these are the 4 amino acids that have a net charge. Okay, so of course we have the CH2, right? And then we're going to have the carbon. But this carbon is going to have a double bond to the oxygen. It looks like asparagine, right? Except you don't have the NH2. Instead you have the O minus. Okay, well, do you know what functional group this is right here? Well, the functional group right there is an ester, okay? And when I think of ester, I think of Easter. And when I think of Easter, I think of chocolate. And when I think of chocolate, I'm thinking about eating it. So you say O8, oh, I ate the ester, okay? So instead of asparagine, it's actually aspartate. So aspartate right there. Also ASP, and for some reason it's D. And of course we don't want any misspelling, so it's aspartate. Aspartate. Okay, so aspartate, ASP, the letter is D, and the tate, or actually the ATE, comes from the ester group. Okay, so similarly we can add glutamine. We can change that, so we can add CH2, a carbon, and then we're going to add a ester group right there. Okay, so instead of glutamine is actually glutamate. Okay, so let's do glutamate is very similar to glutamine, except it's GLU, and then you have an E, right? So that's going to be E right there. Okay, so that's glutamate. So now we can do another one. We can add CH2, 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 and then CH2, and that's the last one, I promise. And we do this. We can add NH3 plus right there. And that is called lysine. Okay, so 4 in the machine for lysine. And whenever you're texting someone and they're lying to you, you can see through their lies, and you put that, well, you're frustrated with them, so you only put K, right? And you know that aggravates people. So when you see through their lies, you just type K. Now for the very last amino acid that you have to know, you're going to add 3 methyls, or 3 CH2s, sorry. Let's just do this right here. Okay, now we get to what? We get to an NH right here. So you can see how similar it looks to lysine. But then it gets even weirder. You get a carbon. You're going to bond this to NH2 right here. And then you're going to have another NH2. But then you have to have a net charge, right? So you're going to add another double bond and put a positive right there. Okay? And that is called arginine. So it might be pretty frustrating. So you might say, oh, you know, so you might say arg, right? And arg sounds like something a pirate would say. So they roll their R's a lot right there. So that's R. And yeah, those are all 20 amino acids, and it was a pain to write them down and to memorize them, but everybody needs practice, so I recommend getting a whiteboard and just writing these out with the letters and abbreviations. So hopefully you found this video to be informative and you enjoyed it. Thank you so much for spending your time with me and learning this material with me. I hope you have a great day and that you're safe. Remember that I will always appreciate and love you. All right, have a great day and be safe. Thank you.